Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We have muted the audio lines, but we welcome you to type in your questions for a speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Marcel Thoen from Wacker Biotech.
Okay, everybody, ladies and gentlemen, it's a pleasure for me to present the progress we have made with our proprietary ESETEC system. But before I will show you the results for the new concept of inducible secretion with ESETEC, I would like to introduce first our company. Well, the Wacker Biotech is a 100% subsidiary of the Wacker Chemie AG, which guarantees a high financial and long-term stability. We are the microbial CDMO with a solid track record and strong customer focus. That means we are able to develop processes for therapeutic candidates from scratch to market, and we have a strong expertise in the production of proteins, live microbials, vaccines, and plasmid DNA. Furthermore, we also offer cost-efficient proprietary technologies such as E. coli-based production system for recombinant proteins and also live microbials. We are operating 4 different sites. You can see here on this slide, whereas the sites in Jena and Halle are mainly focused on the production of recombinant proteins, including feasibility studies, process development, process characterization, cell banking, and GMP production. The site in Amsterdam has strong expertise in the fields of live microbials, vaccines, and fill and finish. But also GMP production of proteins is also possible in Amsterdam and was already successfully carried out for certain molecules. Besides recombinant protein production, we are also active in the fields of nucleic acid-based approaches, and therefore, quite recently, we acquired a site in San Diego with strong background on the production of plasmid DNA. The Wacker Biotech is a full service provider covering the entire process chain from the cell to commercial manufacturing. This includes the steps of strain and process development, process transfer and optimization, and also analytical development. We also offer GMP production of proteins, vaccines, live microbials, and plasmid DNA, as well as release testing, stability studies, and method validation. Our cost-saving microbial technologies include several brands. On the left-hand side, our ESETEC system, which is suitable for secretory production of properly folded target proteins, and this will be the focus of today's presentation. Another brand is our FoldTech technology, also an E. coli-based technology for production of target proteins in inclusion bodies, followed by an in vitro refolding approach leading to correctly folded target proteins. Biotech offers a platform for GMP production of live microbials, and as already mentioned, we also offer GMP manufacturing of vaccines. Now I would like to introduce you our proprietary ESETEC technology, which relies on a well-characterized E. coli K12 strain. ESETEC allows the production of correctly folded proteins combined with a secretion into the culture process. For recombinant protein production, different expression plasmids can be applied that encode for several signal peptides. These signal peptides are a prerequisite for the translocation of the target protein into the periplasm, where then distinct helper proteins, indicated here on the right-hand side, of our toolbox, such as chaperones or isomerases, will assist the proper folding. Furthermore, special fermentation know-how for the different secretion strains allows to steer the fermentation process so that the obtained supernatants will match the requirements for the subsequent primary purification. In the middle of the slide, you can see also, I have to mention that we also offer antibiotic-free selection system, which allows the production of recombinant protein in a complete antibiotic-free way. The clear benefit of ESETEC is shown here on this slide, on slide number 7. The clear benefit of ESETEC is the secretion of the target protein into the culture process. This, of course, reduces the number of process steps like homogenization, IB preparation, and refolding, so that after sterile filtration, the resulting supernatant can be directly used for the following downstream processing steps. And in contrast to conventional periplasmic processes for soluble protein expression, the yields can be significantly increased with ESETEC. That's here shown in the middle. Taken together, the cost of goods can be reduced by up to factor 5 when compared to conventional microbial systems. However, through years of experience, we observed that the secretion efficiency strongly depends on the properties of the respective target proteins. Here on the right-hand side, you can see different scenarios, whereas some proteins are easily released to the culture media, shown in green. Other proteins remain trapped inside the periplasm. To address the different secretion scenarios, different ESETEC strains were developed to ensure selection of the best suitable strain for the target protein, but sometimes such a strain screening only results in the best possible compromise between secretory titer and total titer. On this slide, I would like to show you an example for a really hard to secrete antibody fragment with conventional ESETEC. On the left-hand side, you can the growth and the secretion over time is shown. On the right-hand side, you can see that only a minor amount of the produced antibody fragment is indeed secreted into the culture process. That means whereas the total titer of the produced antibody fragment was approximately 4 grams per liter, the concentration of correctly folded Fab in the final supernatant was only 0.5 gram per liter, indicated with a black bar. One reason for the limited secretion of certain proteins with conventional ESETEC might be the uncontrolled retention time of the target protein in the periplasm. And therefore, the next generation ESETEC with a controlled secretion principle was developed, and as a result, exposure time to helper elements like chaperones and isomerases in the periplasm is now under better control, which optimizes also the correct folding. The controlled secretion is now an additional element that helps to steer the production processes and thus improves the process robustness, which is key to successful process validation and consistent commercial supply of biopharmaceuticals. Now you might ask how the controlled secretion works. It's shown on this slide. The principle of the controlled release technology is based on a decoupling of the production phase from the release phase. Whereas product accumulation is initiated by the addition of IPTG. So you can see the product formation and accumulation in the periplasm. The subsequent release of the correctly folded target protein into the medium is triggered by the addition of a second inducer that leads to the formation of a distinct release factor, which is shown in blue. As a result, an improved host strain is now available, which expands the portfolio of conventional ESETEC host strains for secretory production. This improved ESETEC strain combines an excellent expression rate with fully controlled release of the culture medium. And to demonstrate that, I'd like to show the results on this slide. Here on this slide, you can see now the results for the already known hard-to-secrete antibody fragment with an improved ESETEC strain in comparison to conventional ESETEC. On the right-hand side, again, where the growth behavior for both applied strains was quite similar, now almost 4 grams per liter of the selected Fab molecule was secreted with the improved strain, which, by the way, represents the total amount of the produced antibody fragment. On the right-hand side, you can see the results for a reducing SDS page for better visualization. And as you can see, significantly higher amounts of both chains of the Fab molecule were secreted with a new improved ESETEC strain. Furthermore, both chains were produced and secreted in almost equimolar amounts, and the very low viscosity for the obtained supernatant is of advantage for subsequent primary purification steps. That means the supernatant can be easily processed during subsequent filtration steps such as sterile filtration, microfiltration, or depth filtration. Well, to evaluate the impact of the controlled secretion on the quality of the final supernatant, also the level of contamination with host cell proteins was evaluated by a Western blot analysis, shown here on the left-hand side. The Western blot analysis for a selected cytoplasmic protein, so-called elongation factor EF-tau, revealed that no contamination with that representative cytoplasmic protein could be detected in the supernatants for either the conventional ESETEC or the improved ESETEC strain. Furthermore, that's shown here in the middle, direct measurements for contamination with host cell proteins revealed even a 67% decrease of HCPs per milligram secreted Fab for the improved strain compared to conventional ESETEC. On the right-hand side, you can see also the results for endotoxin, because a key challenge with any E. coli-based system for biopharmaceutical production is the presence of lipid A, also known as endotoxin, which is known to contaminate protein products and cause lethal septic shock. Measurements for endotoxin revealed a similar picture as for the contamination with HCPs. With the improved ESETEC strain, the amount of endotoxin per milligram secreted antibody fragment could be decreased by 71% for the hard-to-secrete Fab molecule with an improved strain. Yeah, and now I'd like to summarize the results for the controlled secretion system. The controlled secretion makes ESETEC a cost-efficient production system, ideally suited for manufacturing of non-glycosylated biopharmaceuticals. It's especially suitable for hard-to-secrete proteins, and significantly higher secretory titers of factor 3 to 8 can be obtained with this new system. The increase of the titer is again protein dependent. We already tested it for several other proteins, and we could significantly increase the secretory titer also for other proteins. The controlled release also allows shorter fermentation times due to active steering of the product release into the supernatant, and we obtain high quality for the supernatant with regard to viscosity, HCPs, and endotoxins, which allows a higher process robustness and a better process control. And with this slide, I'd like to finish my presentation. Here you can see some customers of our customer portfolio, which we support in different processes from feasibilities till process over process optimization up to GMP manufacturing. And with the last slide, I'd like to thank you for your attention, and I think now we are open for discussion and for answering questions.
Great. So, okay. So this is kind of a longer one. So, the question is regarding using a K12-based E. coli strain for secretion of recombinant proteins. How is Wacker's ESETEC technology? So,
yeah, the ESETEC system is based on E. coli K12 strain. Of course, certain modifications were carried out, but I can't go in more detail, which allows, based on genetic modification, to secrete the target protein into the supernatant. So we used an E. coli K12 strain for genetic modification that finally resulted in different ESETEC strains with different secretion behaviors, and finally also in the inducible secretion strain. So the original ancestor strain is a well-known E. coli K12 strain.
Okay. So I've left that question up there. It looks like we're waiting for a few more answers from some of you, so don't be shy. Go ahead and select yes or no. Is antibiotic-free selection important to you? Okay, so the next question is, what secretory titers can be obtained with ESETEC?
So can you repeat the question?
What secretory titers can be obtained with ESETEC?
Yeah, as already mentioned, it's strongly dependent on the target protein, but especially for hard to produce or complex molecules such as antibody fragments, which contain two chains, for instance, the record titers, for instance, are in the range of 7 grams per liter. For other proteins that are not antibody fragments, we obtain titers of 3 grams per liter, and originally the system was developed for the production of enzymes. Enzymes seem to work very well with ESETEC, with up to 15 grams per liter. Another example is irodine, where we have 14 grams per liter, so it's strongly protein dependent, but we obtain several grams per liter in the supernatant.
Okay, so this is a question for clarification. What is ESETEC? Is it plasmid vector E. coli strain, or is it a complete production platform?
It's a complete production platform. That's why it's an own brand. On the one hand side, we have our host strains, we have our expression plasmids. We can apply certain chaperones for proper folding in the periplasm, and ESETEC also includes our internal fermentation know-how for these special strains. So it's a complete package. That's why it's ESETEC. It's Escherichia coli secretion technology. We brand it as a own technology.
Great. So I think we have time for just one more question. If we didn't get to your question, all the questions you ask in the window will be passed to Marcel, and he can follow up with you directly. So if you have more questions, keep typing them in. The last question for the webcast is, do you have to use different chaperone proteins for different target proteins in your system?
Yes, we have. As I said, we have different chaperone and isomerases. And before we start, for instance, the feasibility, we of course ask our customer on the properties of the target protein so that we can also select suitable chaperones and isomerases where we think that these chaperones or isomerases can be of benefit for proper folding.
Okay. Well, thank you, Marcel. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us for future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.